Clarke is immBio's chair and previously its CEO. He was originally a biophysicist, and he's had a long career serving in senior level positions at companies including Smith & Beecham, GSK and GE Healthcare. Today he sits on a number of boards, including the UK's Medicine Discovery Catapult, and more recently he's been a member of the UK government's COVID Therapeutic Task Force. And today Graham is going to talk about immBio's work developing technologies which are able to address pathogen and human diversity. So over to you, Graham. Thank you very much.
Thank you very much indeed. So I will attempt to share my screen, which I hope will work. It's actually very slow at the moment.
It was just working and you seem to have turned it off.
Does that work?
Yeah, it's working. Excellent. Excellent.
Okay. Let's go. All right. So thank you, and a very warm welcome from me as well. And it's a pleasure to be able to share some ideas with you. I've got some slides to share first, but I think often the value of these sort of seminars is in the subsequent discussions. So I'd love to pick those up with everybody. So my first slide is I couldn't help do this. We're in an interesting shaped world, aren't we? And vaccines is really very much at the heart of it. And I think all of us here who've been working in vaccines recognise the enormous potential, but also a lot of challenges. And I suppose the interesting question is will COVID change that? And I think the answer is yes. And the starting position is there's a wider recognition of some fundamentals, which I've put on this slide. I'll be talking mainly about the first two, about diversity, but that's very, very clear, isn't it, from COVID and the concerns about new variants and why some people are getting it and recovering, others aren't, and what's it correlated to and the like. Everyone's really grateful, I think, to have vaccines now available, and it's actually shown you what you can do in terms of speed. Without unduly cutting corners. It may not be quite a normal development pathway, but you can clearly do it faster and yet safely and at reasonable cost when you really put your mind to it. It also shows you that you do need to spend money, and for better or worse, public funding is clearly the main source that needs to do that. But my last point, which I think is where we as a company have very much positioned ourselves historically, and it's even more obvious now, is that you can't be successful on your own, and it's really that combination and that alignment that's really, really important. And we are an ideas generation organisation. We were spun out of the University of Cambridge, but we're never going to be a commercialisation organisation. We're never going to be a manufacturer. We don't have all the specialist facilities that you need for vaccine development, so we can attract a lot and have lots of collaborations. We really value our good working relationship with not just Cambridge, but a whole range of universities. So I think there's some important lessons there that will help us all move forward. So first real slide, I suppose, is a quote from Charles Darwin. And this is often misquoted. A lot of people think he said it's survival of the strongest or the fittest, but it's not. This is the quote. It is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change. And he said that not recently. I had to look this up, the date. It was 1859. So that fundamental identification that variety is generated biologically and really at random, but out of those random variants, some will actually be better adapted and will therefore be better able to cope, survive and procreate. And actually that's fundamentally why you have sex, is to create variety rather than just carbon copies of people. And I think that's a really important starting point for this seminar. So what is that variation? And how does it come about? Well, the pathogen diversity, I guess we understand that things reproduce through various mechanisms, and that in that reproduction, which is quite a complex process, there will be some varieties generated. And out of those varieties, some, as I've said, will be better able to propagate and others won't. And I've certainly been quite angry sometimes at hearing particularly politicians on COVID talking about smart viruses evading and all this sort of thing. Of course, they don't at all. They've got no thinking capability. It self-selects. The other side I think is important, which is the human diversity. And we can see that there are a number of determinants around that, and age is the most obvious one. Immune systems with T cells and B cells, we know that antibody half-lives go down with age. We know that T cells maturation changes with age, etc. So age is, I think, quite well established. Race, sex, you see that a little bit in COVID. Immune status, the most obvious one being where you saw HIV coming along and attacking the very cells that you need for immune protection. And then the last one I note here is the person-to-person diversity. Now, in the immune system, I think we'll all recognise that a particularly important element of that cascade are the MHC molecules on dendritic cells. And when you look at those are both polymorphic and polygenic. In other words, they change an awful lot. So no two people are likely to present in exactly the same way as somebody else in terms of the adaptive immune system. So that person-to-person diversity, if you're taking a Darwinian view of the world, is a good thing. It means that any given pathogen is probably not likely to eradicate the entire population. There will always be some people who are better or worse able to respond to that. So I hope I've set up the case that says pathogenic and human diversity is a big challenge. And now the question is how do you address that? And I will seek to make the case that said multi-protein vaccines, but bear with me and see whether or not I can make the case. So let's just think about the history of vaccine developments. And it depends who you actually ascribe as being the initiator of vaccines. I know there's some debate, but I think in many people's eyes it would be the smallpox, cowpox identification that one cross-protected to the other, and that was a case of a live vaccine in effect. Happened because it was not the same, that it was had a better safety profile, and yet created an immune response. So you can't generally do that. There are very few cases where you can do that. So you have to start attenuating the pathogen. And there's clearly a question as to how far do you attenuate it. The further you attenuate it out, it's probably less likely to revert, but you're tracking further and further out from what you actually need as immune response. So people then said, OK, well, maybe what we should do is kill it. And lots of ways you can kill it. And what you discover is that dead bug doesn't really produce a particularly good immune response system. And actually, biologically speaking, that's not a surprise, because your natural immune system is developed for certain circumstances. It's not developed to have to worry about dead pathogen. So it doesn't have natural uptake mechanism. So you then start saying, I've got to add an adjuvant. I've got to have some way of stimulating, particularly the innate part of that. So you started doing whole kills with adjuvants and then subunits, and then what really is the subcomponent in there and your antigen, through to well, let's begin to conjugate it in some form. So a range of different technologies that I would argue the general direction of travel have been perhaps more highly characterized products. And of course, the interesting one that joins that club now is RNA. Great to see success in there. But the direction of travel, I would suggest, has by and large been from these sort of multi-component products to thinking about specific targeting. But it begs the question about what do you need to put in there to do the targeting, and also how do you indeed make the immune system take it up. So DNA, naked DNA particularly, historically was not successful, again because there was no take-up mechanism. So you had to start thinking of things that would trick the immune system into taking it up. And then you've got VLPs and lots of work on different adjuvants and the like. So that's sort of one way to go. And the other way to go, I think, is which is the way we've gone, is thinking about how do you present quite a lot of antigens all at once, rather than a finite number. And the sort of considerations I think you need to think about is what is it that enables the immune system to respond appropriately, and is your approach actually going to limit the breadth of that response? And particularly, as we know, there are differences, and COVID's another great example that everyone talks about B cells, and for obvious reasons, but nevertheless it's quite clear that protection has a very high T cell component as well. As long as which of these technologies is going to give you the safety and side effect profile, that's acceptable. So you can cut and dice this different ways, but that's perhaps just my way of saying I think the direction of travel quite a lot of the time has been from multi-component vaccines to more highly purified. And I think what I'm arguing, and will argue, is that actually opening it out a bit more actually tackles some hard and difficult pathogens. So, so what do you think about in terms of what you're going to do as a company? And there's a mixture of technical, do I have a solution, and commercial. And one of the things that really has caused us a lot of consideration is about how do you undertake clinical studies? The value in creating a company is not really in the preclinical stage, it's with the clinical. And you won't get traction with large pharma either, by and large, until you've got some clinical data. And that's tricky, because especially when you're into a new disease, a new pathogen, then there often isn't a marker of protection at that point, and so that's a problem. You may be able to do challenge studies, and I know Oxford is one of the leaders in advocating that, which I wholly support. But it too has limitations in that they are atypical individuals from those that you are normally seeking to protect. And the last and sort of the worst is field trials. You know, designing a study. If you really have to start doing, let's take an example. Rationale in the Netherlands had 80,000 subjects in its study for the elderly indication. And that makes it really long and expensive, prohibitively so, which is my next slide, which is, I think, the question is the economics of vaccine development. So historically, it's taken multiple years, lots of people in your studies, and lots of cost. And especially for the neglected diseases, that's really problematic in that you really can't expect commercial backing for a proposition that has that sort of profile. So I guess back to my first question is what do we think COVID will do to that? And I guess I'm rather optimistic that we will see significant improvements in the environment for people like us here in terms of vaccine development. My next slide could be contentious. You can all position it different ways and do different grids, but here I've just thought about, if I was going to plot, you know, how developable are these different approaches, and what do you think the end result's going to be? They're sort of positioned all slightly differently, and what you really like to do is have something that's easy to develop and really good as an ultimate vaccine. But of course, sadly, most of those have been found. It's quite hard to get into that space. So So the next place I want to take us all to is thinking about the immune response, and I think the centre of our world is very much about dendritic cells. I'm the first to acknowledge it isn't the only element, but it is one of the key cells in the cascade. And I would suggest that what we've seen over a couple of decades now is increased understanding about what this important cell is all about and what cell signaling it needs to make it do what it needs to do. And in fact, one of the things we as a company were set up on was thinking about how the dendritic cell normally operates when it sees a pathogen, and how do you reproduce that in a vaccine. Dendritic cells are important in, I would suggest, three sort of broad disease areas. One is infection, which is non-self, and with the right signaling will respond to attack it. In cancer, where you have what was originally your own cells, but it fails to attack it, and autoimmunity, which is it is your own cells, it shouldn't attack it, and it does. As a company, we've focused mainly on infection and to some extent on cancer. Okay, so I wanted to introduce now one of the, I think, quite important components of thinking about how the immune system works. And these have been known for some decades, and in fact, some of the original work went off track, in my opinion, thinking about what these family of proteins are all about. And it's taken some time to really bring them back into clarity about their role. And these are stress proteins. They exist in all cells. They can be upregulated with stress, which is why I called heat shock stress proteins, because that's the most obvious way of regulating them. This is not the same as just turning up temperature to a high level. If you do that, you're going to end up with a heat-killed product. That's not what your body does. It's not what we do in our manufacturing, but stressing is a component. So heat shock proteins are a very interesting family of proteins that have multiple roles depending upon the situation. So they exist inside pathogens, as indeed in any other cells, and I'm mainly talking bacterial here today. I can talk about viral, but mainly bacterial. And their normal role is to act as a scaffolding for folding, because we all know proteins need to be in the right conformations typically to function. So they're folding. So what happens is if you stress a pathogen, the first thing you discover is it upregulates the amount of these proteins. And again, back to biological thinking about why is it like that? What benefit does it confer? I think the answer is it's something to do with locking down the pathogen. So that's some good things. But what it means is that the pathogen now stressed actually contains a higher level of these family of stress proteins, and they're complex with a range of other proteins from that pathogen. And interestingly, when you've generated those, when you now go to the host, the human, what you actually discover is that these stress proteins actually have an innate stimulatory effect, and also that there are specific receptors on dendritic cells that are going to bind these stress proteins, and you find those proteins not only on their own, but attached to the other proteins that they were folding in the pathogen. In other words, they are a component of the transporter route of bridging between an innate response and an adaptive response. So what we do as a company is think about what normally happens in the immune system, and the answer is you start with the pathogen. The immune system kicks in the innate response, cytokine release, etc., and that creates stress. And that stress does something to the pathogen in particular, as I've just talked about. It upregulates components of that pathogen, and actually enables this bridge between an innate response and an adaptive response by being picked up by dendritic cells and chopped into pieces and presented onto MHC molecules will be an T cell response. So if you take that to be a normal immune response, the question now is, can I copy that in a factory, in a manufacturing plant? And that's essentially what we do. So we start with pathogen, which we have to grow. We stress it, and quite a lot of our IP is around how do you stress it, because different bacteria are stressed by different things. You clearly want to kill it, but not in a way that damages the content. And you clearly don't just want a soup. You actually want something that has the most important components in it and not all the debris. So there's a downstream process that again is a very important part of it. And that at a very simple level essentially is how we've approached our technology. And we describe it as a platform because your starting cell line results in a vaccine against that particular pathogen. There are tricks you can play about the cell line and its manipulation. There are requirements to adapt it for that particular pathogen in terms of how does it go through this process. But the principle is that we think we could potentially address quite a few pathogens using this as an approach. And so we started as a company on TB. We were lucky enough to get a decent sized grant from Gates, which enabled us to do a lot of the manufacturing. That took us into a different target, meningitis, and some others I'll talk about briefly in a sec, and then strep pneumonia. So I'd just like to talk a little bit about pneumonia. And I don't know on this seminar how many people work in this field or not. It obviously has existing products, but significant unmet needs, and I'll talk about those to explain why are we working in this field. So I've talked about the technology. So we've got two groups here, elderly and infants we're interested in. We currently move in elderly, and our product essentially is a multi-protein based extract of heat shocked pathogen, which I've already been talking about. So just to put the market context, don't know why the font's changed, sorry. It's always tempting to think about sort of the big three of malaria, TB and HIV as being the big targets, and of course they are, but it's just very sobering to realise that more people actually die of pneumococcal disease than those three put together, and that's despite the fact that there are some vaccines out there which are used. So why is that? Why has it not been fully addressed? And so my next slide talks about some of the challenges. So the way that the current vaccines work is they take the polysaccharide, the sugar coating of it, and they conjugate it and present that. But the polysaccharide is the defining component of a particular variant. So I didn't mean to go there. Particular variant, the serotype. So the vaccine is sort of serotype specific. So if you want any breadth, you have to add them together, which you can do, which is exactly where this technology went to. So PCV 7 is 7 put together, and 13 is 13 put together, and the like. The issue you've got is that the more you add together, you add to the cost, which already is reasonably high. But what you see on the right hand side is that the serotypes that are addressed by the current vaccine does indeed drop, but sadly what happens is that the other serotypes historically have gone up. and you see that both in infants and in elderly. So you are seeing serotype replacement. So an obvious thing to do is to add more into your vaccine, to move beyond a polysaccharide conjugate vaccine, PCV13, to something higher, and people are indeed doing that. It adds to the cost, which is already quite expensive, and ultimately there will be a limitation to how many you can cover. And then the last point I'd like to make on this is of course resistance to antibiotics. So what that means, it leads a landscape with an unmet need, despite big vaccines selling high volumes at high price being available. The next slide just reiterates that slightly different way. It's slightly aged data now, but again it shows you the story of, if you look at these serotypes and you look at 2 time points, then what you saw is again in terms of the blue, the serotypes covered by the initial vaccine has indeed gone down, but the ones that were not covered in it have gone up. And you also see this long tail of different serotypes, and in fact we would anticipate that as you have seen historically, that number will gradually increase. So we don't use polysaccharide, we're using proteins for these reasons here. This is our sort of rationale for moving to protein-based approach, which I won't read through it. But they are far more highly conserved. There's a wide variety of them, and therefore your universality is a much more viable likelihood in a multi-protein based vaccine. And you're looking at a number of intervention points, which I won't talk through here in the interest of time, about things that you want your vaccine to do. It has to do at least one, if not more, of these, which we tested our vaccine to make sure we can. And the model defense is there's a severity and frequency thing going on in terms of consequences of infection, and it's pretty clear as well that how you protect against it does vary as well. So again, it's militating towards a broad response. The next slide is actually, again, just reinforcing something I've already said, which is otherwise normally solid immune system people of a certain age don't tend to get a severe infection. It's the infants and it's the elderly. And the reasons why they're picking it up are different, and therefore perhaps what you need to do in a vaccine is different. But here you see the early age group with high levels, and you see the older age group with high levels, and a dip in the middle. So we need to be thinking about how do we look at the age diversity here in the vaccine. So here's our rationale about the product. We believe that to get the type of response you do need multi antigens in it. We ferment Pneumonia, a particular cell line that we've developed and have genetically modified for specific reasons, and we're doing that, growing it and then stressing it. As I've said already, it is genetically modified to detoxify one of the components, apply D6, which is one of the components that actually causes tissue damage in infection and enables the pathogen to propagate through tissue. So that in itself is a really good thing to be able to lock that up. And I've talked about the process, but I won't go into more detail around that. So where are we on this particular product? We've done lots of preclinical work. We have a cell bank, we have a cell line, we have good toxicology, we show stability. We have a phase 1 study that does what you would want it to do. And we are moving into a phase 2 which we believe the most appropriate is a human challenge study in the elderly, and that will enable us to move into a phase 3 in the elderly. So that's our path. And then nearly at the end, we've applied this approach to a number of different pathogens. We can't do everything. We're quite a small company. We've been lucky enough to get grants to support all of our programs. I've talked about TB. I've said that we've done quite a lot of work on meningitis. We've got good data on two perhaps far more specialist areas, H. pylori, the most common cause of stomach cancer, and C. difficile, the so-called superbug, as well as some cancer work. So we've built a pipeline of candidates, of which the pneumococcal vaccine is in the lead. And so my last and final slide before I sort of hand it over to questions. I've rattled through quite a lot, and I do apologise that I've cut the corners badly on the immunology piece, and I haven't begun really to talk about the manufacturing. But I think this is a sort of helpful statement as well, that whilst it's tempting to copy others and do simple stuff, it doesn't push the frontier forward. And yet, when you push the frontier forward, you do run into lots of challenges, and we as a company certainly have done that. So we're not trying to make life difficult for the sake of it, but our view is that these significant unmet needs in health that still sit out there are going to need bolder, more innovative, more challenging technologies, which we would suggest this is one of them. And we know that it's going to add complexity, but we're not doing it for the sake of it. We're doing it because that's the way we believe we need to go to address the significant unmet needs in the marketplace. So I hope that's helpful. May I suggest that I stop speaking? And I don't know exactly how you want to play it, but I'm open up to questions.
So theoretically speaking, people should be able to unmute now, but the way we do this is you can ask a question two ways. You can put your hand up using the reaction tab along the top. It's next to the breakout rooms and the chat function, or you can type your question. In. So if anybody has a question, either raise your hand, and I'd also encourage everybody, if you'd like, to turn on your webcam so we can see everybody and where you are, etc. So if anybody has a question, either raise your hand or type it into the chat function. So who's here? Can everybody hear me? Oh, there we go. Helen. Yeah, Helen, if you want to turn on your webcam. Oh, and Graham, it might help if you stop sharing so we can see everybody. Go ahead, Helen.
Hello there. Hi, Graham. Thank you very much for the seminar. I'm Helen Fletcher. I'm a professor of immunology at the London School of Hygiene and Tropical Medicine. I'm also co-director of the Validate Network. And, you know, many of us on this call today are interested in vaccine development, in contributing and accelerating vaccine development. So, I mean, I just wondered, you know, what would be the pathway for academics to collaborate with you? So, you know, say, you know, for example, they had some research of relevance, you know, to you, or sort of ideas, you know, to contribute. So how do we go about doing that?
Okay, so well, interesting, you're from London. We do collaborate with Liverpool School of Tropical Medicine. And the reason why we did that and are doing that is that they of course had MRC and Wellcome money to develop a specific challenge model in strep pneumonia. And that is really in recognition that doing field trials in this disease is a really long and tortuous process, and therefore you needed some way of improving your confidence level in your clinical development plan. So what we've tended to do as an organisation is collaborate in specific areas where there's something of particular utility to enable the product development to progress with the sort of confidence that you need to keep investors alongside, and also to be eligible for grants, quite frankly. And so we've used Innovate UK. We've went in with Public Health England to access MRC funding. So we tend to co-apply with an institution. We've had KTN studentships running as well. We don't at the moment, but we have historically. So we've used a variety of sort of collaborative vehicles to keep our business alongside the interests of our collaborator, mainly in the UK, but not exclusively. We've done some manufacturing work at UCL that's got a biomanufacturing centre of expertise. We work with Reading, we work with Bristol. They've got some models as well that are relevant to our work. TB and in fact strep pneumonia enabled through the UK government. We have a joint venture in China with a state vaccine company there. We also have a couple of collaborations in the US. So there's a lot of different models we can follow.
Thanks.
So, any other questions? Bernardo, did you want to say something? You're on mute.
Yes, thanks for putting me on the spot, Blakely. That's very nice. Graham, thank you very much for a very nice talk. I was one of the questions is to what extent are we driving through vaccination? Are we driving the selection of specific strains? And you introduce very nicely in there, also in your introduction, appropriately, the human factor of the variation from individual to individual. So the question, I guess, it becomes twofold. To what extent are we driving to different strains of pathogen, and to what extent are we driving to different populations in humans?
Yeah. That's a really good question. Now, I suppose I could draw an analogue, and the analogue is antibiotics. So what do we know about antibiotics? And the answer is you have a course of whatever it is, 5 days, 7 days, 10 days, 14 days. And the reason why you do that is because you actually want to eradicate the whole of that infection. Because if you fail to do so, the ones that you haven't killed will be the ones that are most antibiotic resistant and will therefore, again, will propagate. And back to my theory, it's not the bacteria is smart, it just happens to be the one that is more resistant. So my personal view is that inadequate protection, sadly, does have a potential consequence of driving serotype replacement, or serotype is only the term for the polysaccharide changes. When you look at the gene sequence, a bit like COVID, there's thousands out there of differences. So sadly, an inadequate protection does have a consequence, I believe, of creating that variation. And I think the same is also true in populations. It self-selects to those who are more vulnerable.
We do see, I work in veterinary species, and I don't know whether you have heard of Marek's disease, in which, so what they were doing is they vaccinated against the original strain.
Yes.
The next strain of virus was a bit more virulent, and so the vaccine, when they prepare it from the attenuated virus, became a little bit hotter, so to speak, in such a way that the vaccine, from being a reasonably safe product, it ended up killing a few chickens, so that you had to make a decision: how do I prevent the killing of my chickens? Do I not vaccinate them and just take the chance or not? So, as a human species, do we need sometimes to sit down and think? Obviously, we want to prevent death, yes, and there is a moral paradox, if you wish, in there. Is that something that is worth exploring or
I think it is. Yeah. I would support that. I mean, it's an interesting thing. If you look at COVID, I mean, the talk originally was that as mutations happen, will it generally get less virulent? And actually, the evidence doesn't support that yet, does it? Far from it, actually. So, dangerous assumption.
Yeah. Thank you very much. Thank you.
You're welcome.
Thanks, Bernardo. So I'm going to give everybody a few seconds now. If you want to ask a question, either raise your hand or just turn on your microphone and webcam and say it. But if not, I think that's going to be quite a nice time to start wrapping up. So let me just give you a few seconds to ask any questions that you might want to slip in quickly. Okay, then. So no, thank you very much, Graham, and thank you for Helen and Bernardo for asking some excellent questions there. I have a quick pitch for everybody because there's quite a few non-validate members in the group in the chat today. So I'm just posting information about how you can join Validate. So if you go and have a click at that, click on that link and consider joining our ever-growing network of vaccine researchers. But with that, first of all, if everybody who can or is willing to, can you turn on your webcams, if you don't mind, because I like to take a picture and I like to have everybody. There we go. Give a few seconds to do that. And I wanted to do two things. If you could all just sort of visually show your appreciation for Graham by waving or clapping, and that'd be really, really helpful because I think it was an excellent talk. So yeah, if you can go ahead and just wave and applaud.
Thank you very much.